Welcome to Microbial Concepts. Today we are diving into fascinating world of extremophiles. Specifically, we are focusing here on isolation of alkali files. So in this video, I will be discussing everything related to alkali files with the practical protocol and Viva questions. So do watch this video till the end. Next is what is alkali file. So alkali means alkaline pH and file means loving. Okay, so alkaline pH loving bacteria are known as alkali files. Same goes for acidophiles, acid loving, so acidophiles. Okay, if you get term phobic, that means fearing, that means it don't like that particular condition. Like hydrophobic, that means water fearing. Okay, so here we are going to uh, talk about basics related to alkali file, and the research paper that I have referred for this particular video is this one. It is a very good paper, and you will find everything related to the practical of isolation of alkali files, okay, in detail. Now the term is used for microbes that grow optimally or very well at the pH above 9, and they cannot grow or grow very slowly at the neutral pH. Okay, so in general day-to-day practicals, you must have seen that we use even the media that are at the neutral pH. Okay, and all the bacteria, they optimally they grow near neutral pH, but here we are specifically giving alkaline pH so that we can isolate our desired microbes which grow very well at pH above 9. Okay, so now these alkali files, they can be isolated from normal soil, but we uh, get very few isolates as we talk about normal soil because the pH uh, is different, right? Now because several microorganisms, they exhibit more than one pH optimum for growth depending on the growth condition, nutrient supply and temperature. That's the reason why we get very few isolates from our normal soil. Now for this particular practical, it is very important that the sampling that you do is from alkaline environment. Okay, so let's know more about it. Now, how do we isolate them as they like to live in extreme environments? So we need to start with sampling, right? Because we need to identify what alkaline environments you have nearby, and you need to get a proper sample, and you use your microbiology techniques to isolate them. Now, sampling is very crucial step in isolating alkali files as we use sterile techniques to collect our sample, and we ensure that we are capturing a diverse range of microorganisms and we are not carrying a contamination with us, right? So that is more important. So what are the alkaline environments that you can go for? So if you have a lake which has a alkaline water, okay, alkaline pH water, okay, you can check the pH of water level and you can use that water as your sample. Next is go for soil samples which have alkaline pH, okay, 9 or above 9. So you will find such kind of soil near industrial plant, okay, industrial area where it is contaminated. One option, or second one is a very arid region, that means dry regions, they mostly have such kind of alkaline pH soil. Then next is cement clean dust, that is actually a byproduct of cement manufacturing plant which contaminates the nearby soil or groundwater. So you can go for samples from a cement plant, okay, nearby cement plant. Then next is seawater. Okay, now seawater, and another example is Lonar Lake, okay, which is in Buldhana, Maharashtra. So you can go for water samples from seawater or Lonar Lake. Now, what is the difference here? Now, both of these waters, they will have some salt present in it with alkaline pH of water. Now, that's the reason why you may isolate a halo alkali file. That means halo is salt loving, high alkaline pH loving bacteria. Okay, so you can go for any of these samples and sample properly, and you can start with your practical. See, This is the reason why I told you you can use the sample of even saline water, because there are two main physiological groups of alkali files. One is normal alkali files and another one is a haloalkali file. Now, what is the difference? Now, alkali files, they require alkaline pH of 9 or more for their growth and have optimal pH for growth as 10. Now, haloalkali files, they require an alkaline pH plus high salinity. That means salt concentration is also required, high salt concentration. So you can see up to 33% of NaCl, weight by volume, is what haloalkali files they can grow in, and that's the reason why I told you that you can go for seawater as your sample. Now, these are few examples. You can go through them if you think you may need for your Viva. Then next is what kind of media one should use as you are isolating an alkali file. So horikoshi or the basal media for alkali file is mostly used, but if you don't have any of these components, you can also go for alkaline nutrient agar. Okay, the same composition for nutrient agar, but you can adjust the alkaline pH to 9 to 11 and you can use it. Now, alkaline nutrient agar, it provides a basic environment suitable for the growth of alkali files while supplying essential nutrients. You can also add few of these alkaline salts and you can adjust the pH. Now, this is the one that is mainly used, that is Horikoshi basal media for alkali files. Now, you can see the ingredients are glucose. Okay, in there are actually two types, one and two. In first, you can see glucose is added, and in second one you can see soluble starch is added. Okay, so you can use any one of it is okay. Now for the first one, glucose, yeast extract, polypeptone, K2HPO4, Mg2SO4 and Na2CO3 with agar is added. And in horikoshi second, you can see glucose is skipped. Other than that, for carbon and energy, soluble starch is added. Okay, and rest goes the same. So you can prepare any one of it. For broth, just skip agar and your broth media is ready. And for plating, solid media, you need to add agar. So now you have the sample, you start serial diluting and you start plating. Now remember that you need to follow all the aseptic conditions while performing these practicals. Okay, so why serial dilution? So this helps us to spread out the microorganisms and obtain individual colonies on plate. Individual colonies on plate, that means you need isolation, right? So it might happen that if you don't dilute, and if you directly put 1 gram of soil in 10 ml of saline and you directly plate it out, you might get a crowded plate. Okay, and it is very difficult to pick up the colony from a crowded plate with surety that yes, you will not get any contamination. Okay, you will not pick up the another colony, right? So we need well isolated microbes, and that's the reason why we go for serial dilution. Now this is the simple protocol. You can see we add 1 gram of soil in 10 ml of sterile water. We suspend it in water. We do serial dilutions. Then we allocate 10 microliters from last three dilutions and we plate it out. Okay, so you can use, you can see here, one drop on horikoshi 1 or horikoshi 2 media. Okay, that means you can use any one of it, and you can then do spread plate, incubate those for at least 3 days or sometimes even 7 days depending on the sample that you have used. Okay, then you may get good number of colonies. Go for isolated colonies, pick them out, you make master plates of those, and then you proceed further. Now, the main thing that comes here after getting an isolated colony is you start testing it for identification. So first thing you go is for colony morphology, then you go for gram staining, then you do biochemical basic tests like catalase, oxidase, etc. Then see for what range of salts your isolate it stands for what range of pH you isolate it stands for, it grows well. Okay, you can check that as well. And after that, you should refer to the Perkins Manual of Determinative Bacteriology and try to identify at least till genus level. And you may also take help for systemic bacteriology, okay, to know more about what kind of the culture is, it is whether it is a bacterial or it is archaea, okay. Then nowadays, next gene sequencing methods are actually used for accurate identification of your isolate if it is a research work. In if you are say this is a college level practical for you, then you do biochemicals and you try to identify it at least till genus level, okay. So this is for the isolation of alkali file, okay. If you just have isolation, then no need of worrying about the identification part. If you have isolation and identification, then you need to go for biochemicals as well and prepare for your viva according to the biochemical test that you are performing, okay, because you will get questions on that as well. Now let's go in more detail for alkali files, because you will get other questions related to the alkali files, like what is the economic importance, why to study alkali files, okay. Now you can see here there are different uses of alkali files. I will list few. So in bioremediation, alkali files, they play a crucial role, and particularly in the treatment of alkaline waste and contaminated soil. Certain alkalophilic bacteria and archaea, they have the ability to degrade organic pollutants and detoxify heavy metals under alkaline conditions, making them valuable for environmental cleanup process. Then second is enzymes. You can see a variety of alkaline enzymes are produced by alkali files. For example, um, alkaline proteases, alkaline lipase, alkaline amylases, alkaline cellulases. Okay, so depending on the type of enzyme, the particular isolate can be used as a detergent in synthesis process for paper and pulp industry, in textile industry, in leather industry. Okay, depending on the enzyme that it produces more. Then next is biotechnology and pharmaceuticals. So alkali files, they have potential application in biotechnology and pharmaceutical research. Their unique metabolic pathway and enzymes can be harnessed for the production of bioactive compounds, antibiotics, and therapeutic proteins. Now, alkaline microorganisms are also studied for their potential to produce novel antimicrobial agents and anti-cancer drugs. Okay, so they have very good applications in pharmaceutical industry as well. Then next is biogas production. Now, alkalophilic microorganisms are used in anaerobic digestion process to produce biogas from the organic waste material. Now, certain alkalophilic bacteria and archaea are capable of breaking down complex organic compounds under alkaline condition, leading to the production of methane-rich biogas that can be used as renewable energy source. Okay, and then comes mining and metallurgy. That is also another application of alkali files. Now, alkali files or alkalophilic bacteria, they are being explored for the potential application in mining and metallurgy process. Now, certain bacteria and archaea, they have the ability to leach metals from ore and mines tailing under the alkaline condition, and this process is known as bioleaching. Okay, so already a video is uploaded on my channel regarding bioleaching. If you don't know the process, if you have this in your syllabus, you can watch that out. The next is alkaline alkalophile bacteria in agriculture. Now, alkali files, they can enhance soil fertility and improve crop production in alkaline soils. Now, certain alkalophilic microorganisms such as alkaline tolerant nitrogen fixing bacteria and plant growth promoting rhizobacteria can form symbiotic relationship with plants and contribute to nutrient cycling and soil health in alkaline environments. Okay, so you can list at least 2 to 3 good economic importance of alkali files, and you can be ready for you. Now about metabolites of alkali files. Now you will think why I have added this in video. Now you will get questions like what is the importance of isolating alkali files in microbiology or biotechnology? Why we study them? The reason is the variety of metabolites that these alkali file they produce. Okay, so they have applications in many industrial fields. Okay, they have applications in food, cosmetic, feed, pharmaceuticals, cosmetic, then biotechnology. Okay, pharmaceuticals, it um, includes all different types of medicinal industries. Okay, so they have siderophores, antimicrobials, organic acids, bioplastic, biosurfactants, exopolysaccharides, other metabolites. You can include enzymes, then compatible solutes, carotenoids, etc. Okay, so they produce different types of metabolites and they have different properties. Like you will find that a biosurfactant has bioemulsifier properties, plus it has antimicrobial or anti-cancer properties. So again, it is used in drug carrying, or it is used as a biomedical material. It is used for emulsifier, de-emulsifying, okay, etc. Such kind of properties they have. Now what is bioplastic? Bioplastic is polyhydroxyalkanoate, PHA. So that is also used in medicine, in biotechnology. Okay, so there are different applications of such metabolites of alkali files, and that's the reason why we isolate them and we study them, okay, for our need. Metabolites that alkaline bacteria or alkali files they produce. Now about the adaptations of alkali files at high pH environment. Okay, they must have adapted to it, right? So let's see what are the adaptations. So membrane structures. Now alkali files they often possess unique membrane composition that enable them to maintain pH homeostasis in alkaline conditions. This may include alteration in the lipid composition of the cell membrane, such as increased level of branch chain fatty acid and changes in the ratio of saturated to unsaturated fatty acid. Okay, so there is alteration in the lipid composition of cell membrane that makes it easy for these bacteria to survive in high pH. Now, this helps in stabilizing the membrane structure and prevent proton leakage. And this is actually crucial for maintaining the cellular integrity in alkaline conditions. Then next is pH homeostasis mechanisms. What is this? So alkali files, they have evolved various mechanisms to regulate their internal pH and encounter the effects of external alkaline conditions. Now, the pH of the cell inside and the environment, okay, in which the cell is, it is always different, and bacteria have to adapt to the pH. So these mechanisms they help, and they may include the production of proton pumping ATPase or sodium proton antiporters that actively transport the protons out of the cell, and as well as synthesis of intracellular buffering molecules such as polyamines and compatible solutes. Now what are compatible solutes? We have seen in the metabolite slide that those are small organic molecules. Okay, now these proton pumping ATPase or the proton antiporters, what they do, they actively transport the protons out, and they try to maintain the pH, okay, inside the cell. And as it is more crucial for the functioning of the cell, so the pH homeostasis is very important. Next is enzyme adaptation. So alkaliphilic enzymes they have evolved to function optimally at high pH levels. Now, these enzymes they often have structural adaptations that enhance their stability, their activity in alkaline conditions, such as increased ionizable amino acid residues on their surface or modification to the active site that accommodate alkaline pH optimum. That is about the enzyme adaptation. Then metabolic flexibility. So alkali fields are often metabolically versatile, allowing them to utilize a wide range of carbon and energy source available in alkaline environments. These metabolic flexibilities enable alkaliphiles to efficiently extract nutrients from their surrounding and adapt to fluctuating microbial conditions. Okay, and last is about osmotic stress tolerance. So alkaliphiles may encounter osmotic stress in alkaline environment due to the presence of high concentration of salts or solutes. So to cope to such kind of stress, alkaliphiles they may accumulate compatible solutes and osmoprotectants, okay, such as glycerin, betaine, or uh, pro- proline to maintain the cellular hydration and prevent the water loss. Now you can see these are the adaptations that alkaliphiles they have over the period of time they have evolved. So you can list at least three, okay, and try to understand the mechanism how they work. Now, some alkaliphiles they may carry plasmid that harbor genes which encode proteins involved in the alkaline adaptation. These protein-bound genes they may confer specific adaptation traits such as resistance to alkaline pH, tolerance to high salt concentration, or the ability to metabolize unique carbon sources present in the alkaline environments. Okay, so you can also check whether you isolate it has plasmid or not, whether the adaptation to alkaline pH is plasmid borne or chromosome borne. Okay, so this is all about our practical isolation of alkaliphiles. So here are a few questions. You can go through them, find their answers, be prepared for your Viva exam. So what are those? Let's go through them quickly. What are alkaliphiles? Why are they commonly found? Where are they commonly found in nature? Why is the isolation of alkaliphile important in microbiology biotechnology? That means what is the purpose? Why to study them? Okay. Then what are some characteristics of alkaline environment that make them suitable habitats for alkaliphiles? We have seen this. Then what are the main steps involved in the isolation of alkaliphiles? So main step is the sampling. Okay. Then comes following all the aseptic techniques, going for serial dilution and the incubation conditions. Then what metabolites are produced by alkaliphiles and how they are useful. Then what type of selective media are commonly used for culturing alkaliphiles? Can you explain the process of serial dilution and its role in isolating? Then what are the some physiological or biochemical characteristics that distinguish alkaliphiles from other microbes? Okay. Then how do alkaliphiles they maintain pH homeostasis in alkaline environment? What are some potential applications of alkaliphiles in biotechnology? Okay, so all these questions I have covered in this video, so you can find your answers and be ready for your Viva exam. So I hope this video is useful to you all. Do like my videos, share my videos with your friends, do subscribe to my channel, and don't forget to comment in the comment box if you like this video, if it was helpful, and if you need any another video for which you are struggling, you are not finding notes on internet. Okay, so I will try to upload, try to help you out. So do reach me out through email or you can comment in the comment box. Thank you.